Well, thank you so much for the wonderful intro. And Emilio, it's good to see you again and pleasure to be here. So, as you just heard, I'm going to give you kind of a summary of the book, as well as a little bit of a sprinkling of some work in our lab at Cornell, where I've been working for about 10 years on some of these projects. And, the book is actually very much a hybrid of those two same ideas, really frontline discussions and ideas of things that we've seen as we get the data from space and as we start to engineer cells to have new features. Then also a very forward-thinking, forward-looking, and prospective approach, and really a hope for what I think will come soon, and the ways that we're building that within my own lab and NASA and with other collaborators. So I'll start first with a bit of background, and then jump into kind of the structures of the book and some fun science tales for the day. So first is, more than ever, we are a spacefaring species. One of the figures from my book just summarized a lot of the work that's done by Matt McKay of, if we look at everything that's ever been launched into space over the past really, 60 years, it's been, clicking along at a decent pace of 50 to 60 or 70 objects, satellites, spacecraft. But really, in the past decade, it's shot up. And last year was the year with the greatest number of launches of things into space. You can see here it's broken down by country. And this is on track to be beaten this year in 2021. So part of this is, low Earth orbit satellites, but some of them are also things to think about. How do we get to Mars? How do we launch spacecraft that go to and from the space station, or eventually a Mars base camp station, which I'll show you today? And how do we think about really using science and technology to enable exploration, including on the red planet? How can we become a multi-planetary species, which again has been talked about for decades, but is now really become much more present, because there is not just governments doing this, and not just two of them. There are now, as you can see from the previous slide, actually a good handful of governments, as well as now dozens of space industries, space commercial entities trying to do the same thing, if not other things. And so that's on one hand, we have this new kind of space race, but we also have, faster than ever sequencing than we've ever had before. This is a plot that shows the cost to sequence one human genome over the past 20 years. You can see something that used to be $100,000 to do one human genome has now come down multiple orders of magnitude faster than Moore's law, which is the speed with which computing power has been increasing and density of transistors. But it's literally the fastest pace of technological change that's ever happened in human history, and we're living through it as we speak, and also benefiting from the fruits of this expansion. So it means it's easier than ever to sequence things. It's easier than ever to go send things into space. And this kind of technology is being used to find new microbes in different places. It's being used to discover new kinds of RNA or new kinds of genes that are found in the human genome. And you might think, well, the human genome, if we finished it in 2001, it was expensive for the first one, but it's getting cheaper. But surely by now we know what most of the genes are in the human genome. And here's what's really interesting. As a geneticist, what's exciting is that we're still discovering entirely new genes. This is another figure from the book, is that in the last 10 years, we found about 10,000 new genes that we didn't even know they were there before. And a lot of them are long non-coding RNAs, you can see. Some are pseudogenes, but these are functional elements of the human genome that we're still discovering today, the fundamental facets that discern our biology and cellular states and how our bodies function. These key levers of function are still being discovered. So it's really exciting that we're using these sequencing methods to find them. We're also using the sequencing methods to find new entities that have their own genes. So for example, how many new species are being found? And you can see here that the rate of it is increasing really quickly and is projected to go very much farther for the years ahead, except for birds. Birds, we've probably found most of the species that exist. They're kind of big and easy to see, but for every other kind of clade of biology, we're still finding new species, entirely new organisms. And this includes, places even like in space. So for example, this is a paper we published finding different kinds of bacteria that could maybe even survive in the vacuum of space. And again, driven by our ability to grab a sample from space, sequence it either in space or bring it back down and characterize it. We've done both and are continuing to do both. And this really gives us this extraordinary microscope, a molecular microscope, with which we can peer into the genetics of cells anywhere on the world or off the world. And what this means is, at least from my perspective, is every day that I wake up, there's actually, more data that exists than any other day before, because so many people are sequencing and contributing and depositing in public records. Every day is actually the best day ever to be a geneticist. So we live in this time of really, what I like to say is genome euphoria, where we can do almost anything and everything with what these tools are at hand to discover new biology and then deploy it. And this is really what's happened in the past 10 years. And so in the first part of my book, I say we've just reached the end of this phase one. And here is the book, The Next 500 Years you just heard about. And at the end of phase one, which I propose to end at 2020, is that we would have a pretty good functional annotation of the human genome. We'd begin outlier studies of humans like astronauts, and we'd be in the early stages of synthetic and systems biology. And so that most of that has actually come to pass. I wrote this in 2011 as the first sort of bullet points of what I think would happen over the next 500 years. And you can see this is kind of the first, and what I plan to live through is the first 50 of it. So I've got hopefully about 39 years left to go. And but what's exciting, I think, now is we really just entered into what I described in the book and in my lab as phase two, is that we're really thinking about integrating new elements into mammalian genomes. This is a picture of Mars. We're thinking about putting boots on Mars. We're thinking about testing the impact of space flight on cells. We're thinking about more astronaut studies. So we're beginning to look at all of these questions, excuse me, in phase two. And one of the places we've also begun to look, as I alluded to before, is even, when we send something to Mars, how do we know that we've sent only our astronauts to Mars? So for example, I've been working with Dr. Katsuri Venkateswaran at Jet Propulsion Laboratory to figure that out. In particular, this is a picture of the clean room where they build spacecraft at NASA. And you can see here people poking around. They kind of have their bunny suits on. They're covered, but they're not completely covered. These aren't hermetically sealed suits. And they're humans, like we are. Engineers are humans too. They might take a little picture with the robot that they're building to send into space, take a little selfie. I think I would do the same thing. But what's exciting about this is, as you probably heard in the news, is that essentially the Perseverance rover is digging up and packaging up little vials to eventually be brought back to Earth. And what it's doing is kind of going around the Martian landscape, looking towards the horizon to find the best spot, orientating itself to dig in the regolith. This is what the regolith looks like up close, and then packaging them up and then putting them into little vials and staring back at Earth. This is what it looks like on Earth when it's about 180 million kilometers away in the sky and waiting for them to be picked up. And if all goes well, in the year 2032, we will have the Mars sample return mission grab those samples in collaboration with ESA, bring them back to Earth, and then we're going to characterize those samples. We'll sequence their DNA, we'll characterize them biochemically, physically. And then one of the key questions that we'll have, of course, as well, for these samples, is there any chance that some of the DNA is Martian? Is it truly an alien? Or is it possible that it could be from one of these guys who maybe had a skin microbiome or some other, little bit of DNA that got left behind? And so part of this is what's called planetary protection. We want to avoid contaminating other planets with our own DNA or RNA or biomolecules, but we also want to avoid them contaminating us. And so, but to really do this, what we need is ideally anything that's ever been sequenced in this clean room to use as an index, and then compare this, for example, to anything that's ever been seen anywhere on Earth, and then compare that to anything we might find on any rock or stone or piece of nucleic acid from Mars. And so what this is the beginning of is this really kind of new era of planetary genomics, where we want to have at scale an entire map of every piece of DNA or RNA or any molecule that's been found on Earth, with which we can compare it to and reference against anything that's seen on Mars or elsewhere. And so we've begun to do some of this work with a project in my lab called MetaSub, which is a metagenomics of subways and urban biomes. It's a nonprofit where we actually coordinate the sampling and sequencing and sharing of sequence information from around the world. This is work with Andre Collis and Gunnar Reisch in Switzerland. And we built actually a graph where you can type in the sequence of anything that's ever been found on Earth, and it'll tell you when and where it's been found. So even if we don't know what something is, at least we can begin to say, well, where has it been seen, which gives us a sense of what it might be doing. And so this project started with me just swabbing subways, but has now become a global project with many other colleagues and collaborators actually doing the same thing. And so this MetaSub data is the beginning of in some of our tools like this one, the Metagraph, has given us a way to map anything that's ever been found on Earth. And we've begun to use this for things like that we find in the clean room. And so we just published this earlier this year, is a framework to characterize organisms for planetary protection. So in particular, we wanted to look at that question I told you about before, is the Spacecraft Assembly Facility, or the SAF, and look to see how does this compare to, say, different controls or ISO clean rooms, and see what do we get if we actually characterize what's in these facilities between different ISO levels of 5, 6, or 8.5. And as a small study, but just to get a sense of what's there. And if you're not familiar with ISO standards, you can see here there's different thresholds for what's the minimum amount of particles or maximum of particle size that you're allowed to bring in. The room air, like, could have mold, spores, fungus, could have, dust, particulate matter. This is ISO 9. But as you get to lower ISO standards, the clean room standards get even more restrict. So ISO 5 is one of the lowest ones we tested. And you can see here, this is kind of what we're thinking is very limited amounts of particles allowed into the rooms. And so what we found is when we actually sequence what's present in controls or these clean rooms, the ISO 5 is actually getting closer to controls. There's very little DNA there as measured by the richness or entropy. These are two metrics that discern what's the diversity of organisms that you find. We don't find that many in ISO 5 compared to, say, the surfaces or the air from the higher ISO numbers, which is intriguing by itself. We also found that there was less DNA and even just less species. This is kind of a coloration of the different species we picked up. You don't see as many reeds being picked up, and you don't see as many species. It's kind of each color here is a different species, and here's the sort of more more permissive clean rooms versus those that are more strict. And you can see here that there's some pretty clear differences between them. But what's interesting is also who we found in terms of the microbes, who was there in the clean room. We compared it to meta subdata in the microbe directory, and we could see for these different clean rooms, some of them even had interesting phenotypes, like they're psychrophilic, so they could survive really cold temperatures, or they form biofilms, or they're radiation resistant, or even radiophilic, some of them. So we could actually see some interesting traits that indicate some of these organisms might not only survive in the clean room, but they might even survive a trip to space potentially, and even there and back. And so this is an ongoing work that we have, is to characterize what could survive there and how do we understand it better over the course of additional selective pressure like space or radiation or even just cleaning. And so this gives us an interesting genetic map. We begin to create this ability to measure and quantify really what are the microbes and the genes that are driving just even survival in pretty interesting environments. And which has been intriguing, but I think this work will be ongoing for years. But eventually, we want to get from not just measuring radiation resistance or characterizing it, we want to begin to think about, could we actually go to actively defending against radiation or damage or enable organisms to survive, in distinct places. We want to move from just measuring and quantifying and characterizing biology and radiation resistance to ideally a more active defense, something that would be a prospective search for ways to respond to radiation and spaceflight dangers. And to that end, that's something I think we'll see, as I describe in the book, as phase 3, is that really begin long-term human trials on genome engineering and monitor and fix any off-target effects. And the reason that we'll need to do this is because if we look at what's ahead, we know that from studies of our own, where we've looked at several dozen astronauts, if you look what's in their urine as they get into flight day 15, 30 or longer, and compared to the days they return or the days pre-launch, we can see the spike in 8-oxoguanosine, which is an indicator of damaged DNA. It means they're literally urinating out sort of the damaged, broken ladders of DNA due to the radiation, which we see consistently higher than controls. And this is just in the space station. This is just in low Earth orbit, fairly, really constrained, fairly safe environment. If you think about Mars in terms of the effective radiation dose in millisieverts, it'd be much higher than what Scott Kelly experienced when he was in space for 1 year. It would actually be, more than even surviving other radiation events. It would probably be, if you did a 3-year Mars mission there and back, it would actually be probably 1,200 millisieverts, which is the hardest trip yet. And so, we might survive, we might be fine, but it raises questions like, what if we could use both physical protection and pharmacological protection? Could we also then use biological protection to keep the astronauts safe? For example, activating genes in response to danger or modifying genes. It raises questions like, could we genetically protect astronauts? And then this, of course, as soon as you think this question, you have to immediately wonder, well, should we do such a thing? The ethics of making humans fit for Mars. And so how do we plan for this? And so we know that, for example, one way you definitely should not do it would be to do it something like what He Jiankui did, where he had a protocol and didn't really inform the subjects and then modified embryos, and then they were born for CCR5. So in particular, I'll come back to CCR5 later, but he basically said, well, I'll get rid of the receptor for HIV so they can't get AIDS, but he may have created new risks at the same time, which I'll talk about in a little bit later. But we know this is one way you don't want to do it, is like top secret embryo engineering and then release it onto the world. He's now in jail. But instead, you want to make something much more open and collaborative and engage the community. This is something we're doing with the Genome Project Right Consortium, is to think about how do we modify cells and get them out into patients therapeutically? How do we understand the ethics, the technology, the implementation of this? And this might sound almost alien to say, well, can we modify human cells and put them in back into people? But it's actually already happening now. And so, in particular, we have set up multiple clinical trial sites for CAR T trials, or chimeric antigen receptor T cells. And if you look at what these are T cell therapies where you actually take a T cell, modify it, and make a chimeric antigen receptor, and then you put it onto the T cell. You grow these T cells up, infuse them into the patient, and these T cells then bind to the cancer cells ideally and kill them. And then eventually, we can actually even reprogram the T cells if needed later on. And so this kind of technology is something that's being deployed, not just in one or two sites, but actually now dozens of clinical trials are using CRISPR or modified versions essentially of these T cells. And there's over 1,000 clinical trials just doing some version of CAR T or CAR NK trials. And so we know that this is an ever expanding list of sites and places that are actually doing this sort of extraordinary modification of cells, using them therapeutically and putting them back in patients. And so if we can do it there, it raises the question of, well, could we do it elsewhere? So for example, what are other things that we could modify? Could we try to do something like, for example, P53, which is a gene that is the guardian of the genome and scans for DNA damage? You can see most organisms have two copies or one copy of this gene, P53, but some actually have, you can see, who's this one outlier here? An African elephant has 20 copies of the gene for TP53, and they have a much lower risk of cancer. So could we take a lesson from them and maybe do it in human cells? Some preliminary data shows that that's definitely possible. But there's other creatures that also have really intriguing and distinct ways to resist against radiation and risks of space. That's one of them is a tardigrade, these cute little organisms called the water bears. They can actually go into the vacuum of space and survive, and they even come back down and be rehydrated and start walking around. And so a group by Hashimoto et al. Actually noticed in 2016, they actually got one of their proteins to show that it could help human cells withstand radiation just in a temporary fashion. So inspired by the Hashimoto paper, we've actually now done a stable transfection of cells in the lab. And we can see that we have, really constitutively expressed DSUP in human cells. These are HEC cells. But here, after radiation, you can see 1 gray versus 0 gray of radiation. The DAPI are the nuclei of the cells. DSUP here in green, you can see where the DNA breaks are. You can see DSUP quickly moving in to help expedite DNA repair from what we've seen so far. And so what's really kind of exciting is it's not just a little bit of help, it's actually giving an 80% reduction in 8-oxoguanosine. It's that same molecule I talked about before, where the amount of reduction of this you can see here compared to our empty vector or to the control cells, both very high. And this is the negative control here. We can see we're getting this massive reduction at 1 gray, 2 gray, even less of it before the radiation starts. And then once you get to too much radiation, then it breaks down to 0. But we know it's possible to use genetic engineering methods to actually improve the reduction to improve the ability to withstand radiation and survive just fine. And that's just at the genetic level. That's something I think most people have probably heard of. You think of ways to do genome modification or insert genes into genomes. These are things that people have probably read about or heard about for a while. But there are other things we can do to the genome. So for example, we could use epigenetic methods for CRISPR, where we actually, instead of thinking about the canvas of life being just a genome, you can turn genes on or turn genes off as another means by which to control the functions of cells, to control what genes are doing. So in particular, multiple methods have been published now that do this. One of them that we use in the lab now is a deactivated Cas9 system with TET1. There's other versions where you can add a methylation group or take it away that have been published by Charlie Gersbach and others, is that you can actually add methyl groups or acetyl groups to your histones to modify them. You can guide cell differentiation or reprogramming and study different therapies. And so this field has really just gotten to explode. And one of the bits of work we've just started is working with DARPA to think about this preemptive expression of protective alleles and response elements. And this is called the PREPARE program. Is could we actually stitch together a series of functional tools where if you're getting into a radiological hazard or a biochemical hazard, to activate the genes before you go into that risk area. And so we're thinking about doing this, first in human cells, but eventually this is something that could be deployed therapeutically also for anyone, soldiers. It could be for medicine. It could be for people really encountering any risk of any kind. And so this is really, in the next 30 years. But if we look out a little bit further into the look ahead, eventually in phase 4, I think we'd start to test these protected human genomes, in space environments and figure out, can we see how well they do and monitor them over the long term. And when we start to get to these environments, we would have very limited resources. We would have, only what we'd bring with us, very limited ability to culture anything or to grow anything on these planets, including Mars. And so what I started to think about, and wrote about in this part of the book is, could we remove what I like to call molecular ineptitude, things that we should be able to do, but we can't. And so one of my favorite examples of this is, for example, vitamin C. We cannot synthesize vitamin C on our own. The gene is still inside of us, but why not bring it back? We have an ability to make vitamin C, but it's been lost. And so this is not just true for humans, but you can see this biochemical pathway for synthesis in bold. You can see it still exists in dogs and cats and sheep. They don't get scurvy, but you can see it has been lost multiple times throughout the primate and also mammalian lineage, where they just no longer can make your own vitamin C because it was enough of it was present in the diet. But what if we could bring this back and just reactivate the pseudogene? I mean, we could do that. That's something we just started working on in the lab. But the other thing we've been working on as well, it's also we have to get 9 of our own essential amino acids from our diet. Why? Why can't we make all of our own essential amino acids like some bacteria can do or some yeast? And so we know organisms that can do it. We can actually learn from them and actually do this. So, for example, what I created here a bit in the book is different genes from other organisms that if we stitch them together into the biochemical pathways for humans, we could, in principle, actually make all of our own amino acids as a human cell. Then eventually this raises really big questions. Well, if we can, add and copy and modify and really reconstruct different parts of the human genome, could we repair disease genes? Could we make it so we no longer have any of these, high risk disease genes that are present, especially if you're far away from Earth, you would want to avoid any disease that you could. And this is actually, again, there's evidence of this already in the literature. So, for example, some of the genes for hypertrophic cardiomyopathy, which can lead to heart attacks, have already been fixed in embryos. And this here by Shurkat Metelapov. And you can see, it's been done there already for embryos. There's other variations of this idea. What if you have a mitochondrial disease? Could you actually have, an ability to have a donor from the mitochondria? Or what if you want to have 2 mom and 2 dad children? This has been done in mice. You can have two mothers actually reproduce just using 2 sexes, 2 men or 2 females. Again, only been done in mice, but there's no reason that wouldn't necessarily work for humans. I mentioned already there's the idea of having 3 parent children. So that's having 2 parents of the same sex. You could have 3 parents of 2 and then one also of 2 sexes. And that could be, 2 female and 1 male here in this case. And so this idea of having, what I like to say is an opening of what is, the definitions of who can be a parent and who can contribute towards a parent has been really broken open in the past 20 years of how do we define this? What does this mean? And even the ability to raise a child or to have it come to term has been also dramatically changed by technology. So, for example, work on extrauterine systems, or what are called exowombs, have been really pioneered in the past 5 years especially, to actually grow a lamb from extreme prematurity all the way through to adulthood. In particular, you can see here, this is kind of an exowomb where you can grow, and you can see the lamb continuing to make its way through the rest of, essentially embryogenesis, and then until it can survive on its own. And so this really raises interesting questions. What I think about this when I look at such images and read such papers is I think this means you could actually have cellular liberty from the very first cell. As soon as you get any cell, you no longer have to be subjugated to the whims of the genetic shuffle that it was handed. You can modify it, you can change it, you could really describe and activate your own fate. You could potentially avoid disease, you could potentially have additional features, and you would not just be, you'd have total control over your own cells, really for the first time ever. But this does really raise really big ethical questions, like even just simply on a technical question, outside of whether we should do such a thing, which is not clear, or currently the answer is probably no. But even just, can we really select the best embryos? Do we even know how to do this? So this has been described in literature by many people who've really critiqued these ideas. And in particular, here's why. The reason is it sounds pretty straightforward. If you think about a polygenic risk score, this is a risk score for any different disease. It's the sum over m different genetic loci, these genetic markers across these markers, and the weights for any effects of a given marker for a disease. The way this generally works is you take this polygenic risk score, or PRS, you look at cases of people who had the disease, like, for example, cancer, breast cancer, controls who didn't get breast cancer, look at the genes that give you a high polygenic risk score versus a low risk score, and you optimize for what would give you the lowest possible polygenic risk score. So it seems pretty straightforward, it seems laudable, seems like a good idea. But the problem is, imperfect measures can give you imperfect metrics. So in particular, there's a bias to this prediction because the ancestry itself of your DNA can actually change your polygenic risk score prediction, because your risk score, for example, for schizophrenia is more associated with ancestry than it is for actually for schizophrenia. Work by Francisco de la Vega and Carlos Bustamante and others have shown, this is a key factor that has to be understood. And so in particular, you want to think about if you're selecting embryos, it has to be not just what we know at the time, it has to include accounting for ancestry. But even that is too limiting. I would argue, and have argued in the book, that the risk score for any given disease and treatment in a given place, including in a new planet, depends on a lot of factors, many factors, not only just in the individual, but what happens over the course of many generations. What does the heritability look like? What is the pleiotropy? This is when one gene can have multiple functions. As I alluded to before, CCR5 has more than one function. So if you decrease your risk for HIV, you'll increase your risk actually for West Nile virus. So these are all factors that I think should all be part of this consideration of what is your real risk score and what happens when you utilize changes to it. And you might think, could we have a multi-generational genetic experiment or a mapping and monitoring of changes of allele frequencies? What would that look like? Again, it almost sounds like science fiction, except that we know some of this can be done because it's already been done. In particular, in 1959, what began the silver fox domestication experiment. There's a really wonderful review article here about it. Is where what they did is they took, basically different wolves and said, okay, let's just take all of the wolves that are the most aggressive and let them just get old and die. But those wolves that were the most tame and sort of the most playful, let them reproduce only with each other. And so this began after multiple generations. And actually, by 1969, this is one of the first foxes that actually started to have floppy ears. So as you took more and more tameness as an actual phenotype and did directed evolution, eventually we got floppy ears. They had more juvenile characteristics, like a shorter, rounder snout than other foxes. They began to look like dogs. And what's interesting is they also eventually had changes in their brains. They took, silver foxes here, again, got rid of the aggressive foxes, let them die, and only went for the tame foxes. And when compared the prefrontal cortex to the tame versus aggressive foxes' brains, even their genes had changed. What was different in what's being activated in their genes for the tame versus aggressive from their brains. So we know that actually a directed evolution over the course of only a few decades can actually pretty dramatically change not just the phenotypes of your organism, but even the brain chemistry and brain functioning that's happening inside the animal. And so it's a really extraordinary experiment that shows that it is indeed possible to do in a relatively short time frame in a mammal. And so this raises, questions of what we could do looking further on directed evolution. But eventually we might have to think, well, could evolution allow us to survive, for example, on Europa or these really far away alien planets? And this is where we get into phase 5, where I think we might have to get even more creative about how we would enable survival on different planets. So in particular, describe, the ability, could we create new whole genomes from scratch? Like, could we synthesize DNA and make a whole new genome? And the answer we know is yes, at least for E. coli. There's been an entire synthesis and creation of E. coli you can see here. So we know it's possible and that these tools will get better. But eventually you wouldn't just want to say, let's synthesize a new genome from scratch, but could we add whole genome components, whole mitochondrial variations, or whole new organelles, whole new pieces of function inside cells? For example, what if you could add a chloroplast into human cells? Could you have chlorohumans where you get green humans that can harvest skin? So I did a Q&A about this a few years ago and then did the math on this to say, well, let's see, say you wanted to go out to the pool and just lay out your skin and say, oh, I'm feeling hungry, I'm going to lay out my skin and I'll get full. How much skin would you need? Well, it turns out, based on some basic math, if you lay it outside for 1 hour, you'd need about a 300-fold epidermis expansion. And then what that means is it's about the equivalent of two whole tennis courts of what you'd need for the expansion. So, interesting thing you try to fold up, but certainly shows that this is actually possible. The other thing that shows that, we know it's possible to start to think in these lines is chimeric embryos like human pig embryos or even variations of hybrids between primates have already been shown that it's possible. Again, it's not that it's easier, that it always works, but we know that it's possible, and more of this is happening as we speak. The other thing that's happening is our understanding of genomes has changed. Like this seems peculiar. This seems unnatural. It seems odd, but it actually isn't that odd. So, for example, other organisms have been called an evolutionary scandal, like bdelloid rotifers, because they have a life sometimes without sex, which sounds awful, but they do it because sometimes they can get borrowed DNA and use it for their own purposes. Like 8% of their genome can be foreign sometimes. which again sounds weird, sounds odd, except for the fact that even the human genome is probably from an old dead virus that's embedded into our genome. And so even us walking around as humans, we too are relics of what our genetic past has been, as well as kind of our ongoing genetic changes. So there's an ongoing effort clearly in our bodies. And so this means that I think this mix and matching of functions and cells and genomes is something that to some degree is already happening, which we would just be doing a lot better and with more volition instead of doing it just by accident. So that then leads us towards an interesting future where eventually we start to think about how could we get to and survive on these really extreme environments. And here we take lessons from a project in our lab called the Extreme Microbiome Project, where we profile novel and extreme environments with genomics methods and learn, well, how do organisms survive in places with really extreme salinity, temperature, pressure, moisture, toxicity, or pH, and take those really as substrates to learn to apply them essentially to other organisms. And so this process has just begun now, but by the time we get to 100 years from now, 150 years from now, we would have even more of this data and eventually even have it from other planets if all goes well, including Mars, if anything survived there. And so, again, we can find them in things like the space station, and they can actually give us new molecules that could be biofilm, sunscreen, and lead to DNA repair. So we know it's possible to find weird things in creatures, even from the space station. And so looking out then a couple hundred years, the hope would be by the end of, say, 2300, we'd know enough of this to begin to seed new genomes to on new Earth-like planets. And so the goal would be by then Mars would begin to look something like this, where we're going from just a handful of people, transient crews, to maybe hundreds to thousands of people. And we'd go from a very minimal ability to have self-reliance, but eventually begin to make our own what's called in situ resource utilization, where we could actually begin to build all of our templates for what we'd need essentially on our own and have Mars be self-reliant. So this would take, again, likely a couple hundred years to get to this stage, but the hope is by the end of the stage that we would be there. And then once you could actually go to Mars and say have a pint and kick back or meet friends, we would begin to have as a species planetary liberty. And the idea is the same as cellular liberty, is that can you do anything that you want with your cells? Can you analogously go to any planet that you want to go to? Is an extension, I think, of the liberty of a species, that you have the freedom to go visit a planet, which right now we do not have that freedom. We can barely survive anywhere outside of Earth. But what's exciting about this is I think by 2300, we'd reach that stage. And then around that time, we'd begin to say, well, what other planets could we go to? We'd begin the selection of new candidate worlds, in which, again, in the early 90s, we had only a handful of planets, of exoplanets that have been discovered. But now we're finding multiple planets usually per day. And you can go to this website and look at all the exoplanets that have been found. There's actually thousands of exoplanets that have now been established. And there are hundreds of them that are potentially habitable. Here's an example of some of them that are Proxima Centauri b or TRAPPIST system. These are, again, 20 years ago, we had no human genome, or we just gotten it, no beginning of the human genome, and very few exoplanets. Now we have thousands and thousands of genomes established. We have thousands of exoplanets. Some of these look like they could be habitable. And a really fun part of the book was putting together what do we know for the different planets and how similar are they to the Earth. So what is the Earth similarity index? If you look at Earth here versus Mars, and within the first 5 parsecs, what's the temperature of the planet, the density, the solar flux, and other physical characteristics of the planet. Ideally, if all these are full, you get an Earth similarity index of 1. As you get more of them full, you get an ESI above 0.9, which is blue. If the ESI is above 0.8 to 0.9, it's purple, and other ones below that are kind of gray or white because we don't have no data for gray. But you can see within the 5 parsecs, there's actually a fair amount of blue planets that are not too far from Earth that maybe we could get to and survive on. And if you go out to 15 parsecs, you can see there's a whole bunch of other planets that we've already found in the past 5 to 10 years that we could get to and maybe actually survive on. We wouldn't, no guarantee till we get there, but it's an extraordinary to think that they're not that far away. And if you look out within our sort of galactic neighborhood in 20 parsecs, there are some that are 1 or 2 light years away, some that are dozens of light years, but all relatively close. And if you zoom way out where we are in our galaxy, here's the Milky Way, here's the side view, and here's a view from the top. You can see these are all the exoplanets that have been discovered across 3,000 and 5,000 parsecs. And you can see all these planets here in one big line because that's where we've been spending most of our time looking. We haven't even scanned the rest of the sort of local galactic area in this quadrant, but we could, and we can already see just from some transit data that there's other good blue spots here and purple spots, places with a pretty high Earth similarity index that we could get to and maybe survive even in our current form. And maybe one might not need as much genetic engineering, or maybe we just need a small tweak to survive, and that is very possible. And so my hope is then basically when we get towards the end of phase 9, that we'd understand enough to how to send humans to go towards another star, that we would launch shipments of humans to new worlds. We basically call this a generation ship, where multiple generations live and die on the same spacecraft on their way towards a new world. And so together, in the book, I described this as a launch from what's already been established in the past 70 years for evolutionary technologies, genetic technologies, cellular engineering, transplantation, regenerative medicine, developmental biology, and how society has changed. There were forced sterilizations that happened into the 1970s in the United States. Unfortunately, they have stopped. This is the opposite of cellular liberties, taking away people's rights. I think we're finally getting back towards where you can actually enable more rights with people's cells and with their own bodies. And so the hope is that by the time we get out towards the future, all of these different threads of technology and society and development would get us to a place where we'd have actually a virtuous cycle where genetic diseases are solved and polygenic challenges are also corrected. You could even have autotrophic human cells, or you could have directed evolution for Mars, and you could have this accelerated directed evolution for space exploration, even the creation of new organs, or point-to-point biology, where people see an organism on Mars, understand it, sequence it, send the data back to Earth for synthesis for further study, and begin to actually have evolution being modeled and understood across multiple planets, and hopefully eventually entirely different stars. And so I think this is kind of what's projected of what I think can and will occur, over the next, basically, 500 years. Early at this point, over the next 489 years to go. And you can see here, this kind of the summary in the book. And so I get a lot of people will say sometimes, well, Dr. Mason, that's interesting and fascinating, but why do all this? What's the point of actually planning this out, and won't we all be dead? And what's the point of it anyway? And also other critiques, like, shouldn't we also solve poverty and crime and other sociological or structural injustices that are in our society before we do these other problems, these other challenges, and address those? But we can walk and chew gum at the same time. We can do both things. In the United States, for example, we passed the Civil Rights Act to enable better voting and less, racist policies, while getting people onto the moon. We could do that at the same time. And also, there are other more substantive reasons as to why we should do this. And so I describe this as a kind of an ethical duty. And it's because we know that life is fragile. We know that, for example, in North America, 1 out of every 4 birds is gone in terms of species over the past 50 years. And a few of them have increased in numbers, but vast numbers are gone or severely diminished due to basically disruptions of their climate. And this is not the first time that this has happened. If you look over the past 500 million years, in a world both with and without polar ice caps, there have been 5 mass extinctions you can see here, where the temperature of the Earth has fluctuated between 10 and 32 degrees. And every time that there's a mass extinction, it's when there's a large scale change in the temperature. Lots of things die. And so we know that anyone from 60 to even 96% of species have been killed in these different mass extinctions. And the big worry is that we're creating a new mass extinction event today, which some call the Anthropocene, based on what we're doing to the Earth today. But this world, while it does seem to fluctuate and cause extinction, is actually, if you zoom out a little bit more, is really this interesting place in history where we can see that there's been liquid water on the Earth for the past, several billion years, but there's been times where there's a global cooling multiple times. Now we're in liquid water. But if you actually look at where we are, it is a nice, but a very temporary temperature canyon. We know eventually that there will be, you can see the sun will start to heat up, the oceans will start to boil, and it just means we cannot stay here forever. And so if you look far enough ahead, all the creatures of Earth are at risk, ourselves included. So we used to think of food in general like this from elementary school. I was taught that there are producers and consumers and decomposers, that there are just 3 kinds of organisms. But I argue to you today that there's actually a 4th kind, and we are a unique kind of species. We're the only ones that are really aware of extinction. We're the only ones that can guard against it. So only humans are extinction aware. Only we can therefore prevent it or mitigate it, and even in some cases bring species back from the dead, like we are doing with the woolly mammoth right now. And this is really a profound responsibility that, for example, as species get low, we've begun to work on the Revive and Restore project and sequencing the genomes of these cloned ferrets to actually make sure that they're healthy, and begin to do this not just for species that are at risk today, but I think we have a duty to all life, both past, present, and then eventually the future life that we haven't even found yet. And this really gets us, eventually, We keep doing it on this planet. Hopefully we do it on Mars and other planets and serve as really the guardians for life forms. I argue this should be a purpose, really a duty for our life, our genetic duty, or I call it deontogenic ethics for what we need to do towards other creatures, including ourselves. And then you might say, well, okay, fine. If you keep going to one star to the next star, eventually, will the universe end, or will the universe collapse back on itself? Will it rip itself apart? And I think the biggest ethical question at the end is, well, should we stop the universe itself from being destroyed because we're worried that life wouldn't arise again? And here I think we should do what we've always done as life forms, is actually just survive. I think we should actually prevent and even restructure the universe so life can survive, because we might not get life again in the next universe or any universe again. So it's a really rare and precious entity that I think calls upon us to serve as its guardians. And so that's how I close the book. That's how I'll close the talk. I want to thank everyone who's made a lot of this work possible, particularly a lot of the people from the twin study, collaborators and astronauts like Mark Kelly and Scott Kelly, everyone in the laboratory who makes this work possible. These people are awesome. This is a lot of them post-vaccination, also feeling better. So really fabulous group. Thanks, of course, to funding from NASA, World Quantum Gates Foundation, and the NIH. And then also everyone, our friends, colleagues, collaborators over the years, all of whom have made this possible. And I couldn't have done any of this work without them. So happy to take any questions, and thank you so much for your time.